Hello everyone, thank you for coming. It is my pleasure to welcome our today's speaker, Professor Boris Shraiman from University of California, Santa Barbara. He's a very distinguished speaker. Let me just very briefly tell you his career trajectory. Boris has received his PhD from Harvard. After that, he completed his postdoctoral training at University of Chicago and in Bell Labs, where he then stayed as a permanent member of technical staff at the theoretical physics department for 16 years, and has done research in various topics such as correlated electrons and superconductivity, pattern formation, and turbulence. While at Bell Labs, he started already to think about biological problems, but then started to focus on it completely when he moved first to Radegers in 2002, and later since 2004, he has been professor at physics department at UC Santa Barbara and a permanent member of Kavli Institute for Theoretical Physics. His lab focuses on morphogenesis and statistical genetics. What it means you'll learn today, especially we'll get a beautiful canonical example of morphogenesis, the fly morphogenesis today, the lecture on the physical side of it today. So I'm very much looking forward. Please, Boris.
So thank you very much for inviting me. I guess I haven't been here for a little while, so I reported for duty this morning to Mayor Hall. And thank Alexander for the board introduction. I myself would have introduced myself just as Pierre Hornberg's postdoc. So, anyway, well, I'll be talking about morphogenesis. So, as far as colloquia go, this would fall into the category of physics of living matter. And so physics of living matter is this discipline between physics and biology. And quite generally, you might wonder, what is it that physics can offer to biology and vice versa? And well, one obvious thing is biological phenomena do deeply involve physics. I would call this biophysics in strict sense. Biology is using, of course, physical interactions and instabilities to do its business. And not to be overlooked, so a very important contribution of physics is actually developing all these tools and the instruments that our biology friends use. But physics also contributes, sorry, I'm going to rearrange the physics has another contribution to make, and that's in the way we approach our studies. In physics, we actually are quite fond of phenomenological models, which are rather foreign in biology. And I think there is an impact for our field to make there just by making phenomenological modeling sort of an acceptable practice rather than a bad word. And then I hope to convince you here that at least in some small way, when you think about biological phenomena, you can encounter some bits of physics that you actually did not learn from Landau missions. And perhaps we can also try to formulate some questions that we should be thinking about, even if we may not know an answer. So let me start with this biophysics phenomena in the strict sense. So the amazing thing is that we, broadly speaking, keep discovering new effects. So one of the exciting developments of the last 10 years is the discovery of what's called membranous organelles, basically liquid droplets phase separated inside the cell, and by virtue of their different compositions, they selectively partitioned biomolecules and therefore localize various important biological functions inside the cell, right? I guess I took these pictures from Cliff Branglin's review paper, but I could have used pictures from Alexandra's papers. So if you want to learn more about this phenomena, you talk to Alexandra. I zoom right on to the next little example taken from the work of Maha and his friends well, this is just one particular nugget from a large body of work exploring the effect of different mechanical instabilities in morphogenesis, in the development of animals and plants. So in this case, you're looking at the chicken gut, which is an intricately coiled tube, and Maha and friends illustrate that you can understand the shape of the gut simply by realizing that it's an elastic tube glued to a strip of elastic tissue with different properties, and that in itself is enough to, okay, there is mechanical instability leading to formation of these countercoils, and this provides with enough understanding of the phenomena that it is actually predictive theory. So one can relate the relevant length scale here to the properties of these two different types of tissue. And that's, I guess, sort of the first little nugget. So again, a very simple model, very simple physics, but it is predictive. So it can help you understand how one observable depends on some other property. So this was an example of physics in morphogenesis, and at least intellectually, this whole field goes back 100 years. Darcy Thompson actually thought that all of developmental biology it should be thought of sort of mathematics and physics. He was just observing beautiful shapes in nature and wondering how they might be described, and had some very beautiful ideas. But he was really much before his time, in the sense that at the time when he wrote his book, I guess genetics was just emerging as a field, and developmental biology wasn't quite, well, it was also emerging as a field. So 100 years later, wouldn't it be fun to go back to that Darcy Thompson's agenda and revisit it with actual knowledge of molecular genetics and developmental biology that biologists have given us since. For me personally, at least, a little nugget question one extracts from that is, how do genes encode geometry? So how do we program the developmental process so that at the end of the execution of the dynamic program, we end up with some non-trivial shape? So Darcy Thompson was talking about growth and form, and here is a very simple example in which growth actually controls form. In this shape, in this case, we're talking about shapes of leaves. So you know plants have leaves of different shapes, and it was demonstrated that differential expression of a certain particular gene, doesn't matter what it's called and what exactly it is sufficient to change the shape of the leaf. And what the gene does is it controls the rate of cell proliferation and growth in the leaf. In such a way, so actually I guess this one represses growth. So now if you, okay, yes, this is a real leaf, and this is a real cartoon of a leaf. Right, the spherical cow approximation, spherical leaf, no, excuse me, it's not spherical, it's actually a disk. So 2D. Of course, if the growth of this little disk is uniform, it's just going to expand as a flat disk. Now suppose growth is, it grows more in the center. So in order to accommodate those extra cells, the disk will want to buckle out, and effectively by controlling the rate of growth, you're controlling the Gaussian curvature of the embedded surface. And if you do the opposite, if you suppress growth in the middle, then you generate negative curvature. And that's exactly what happened to that leaf. It was overgrowing on the periphery. So by controlling growth, we can control the shape of these embedded surfaces. And here's an example of the ideas flowing in the opposite direction. So several groups actually, this particular paper comes from the lab of Eran Sharon in Jerusalem. So demonstrated that by generating concentration gradients of certain polymer, and then subsequently curing this polymer in the hot bath, they could generate sort of differential swelling in this gel, and again control the shape of this embedded surface. So, and there's a whole little subfield of these sort of programmable materials, which in part is driven by these ideas. So, in this little illustration, we see the direct connection of form, so the geometry, to mechanics. And so if we want to understand morphogenesis, we should be thinking about mechanics. But differential growth is only one of many aspects of mechanics involved in morphogenesis. And let me show you another little example of a morphogenetic process, in this case involving the earliest stages of fruit fly life. So embryonic development. So in this movie, you'll be looking at fluorescently labeled nuclei that are arranged on the surface of the ellipsoidal shell, which comprises sort of the earliest stages of fly development. Maybe I'll tell you right away, you will see these, so all nuclei here are labeled in red, but there is also a colorful reporter for some of the cells or nuclei here that are expressing one particular gene. And what we will see is that, so at the earliest stages, the embryo just consists of so several thousand of identically looking nuclei packed on the surface, but they're not really identical because some of them are beginning to express different genes. And there's a whole wonderful story to be told by Claude better than me, of, now that was discovered by developmental biologists, of how the spatial pattern of gene expression comes about. But that's not what I'll be talking about. I'll just show you what happens to these cells, or nuclei in this case, once they start going about their business. So, right, we start pretty much with all cells, all nuclei being the same. Now, some of them started expressing certain important developmental genes. And I should stress that this is only one of the many patterns that are programmed into the embryo at this stage. And the next thing that happens is cells start moving about. And the first thing they do, first thing that you see here is gastrulation. So you're looking at the ventral side of the embryo, so you're looking at the belly, and cells start moving towards the ventral line, and then they dive inside and form the inner layer of tissue. And then lots of other things happen. So cells are basically moving like crazy on the surface, and this image comes from light sheet microscopy, so we're actually seeing both the ventral side and the dorsal side, and all the sides are available in that data. So there is this massive rearrangement of cells that is happening very early in the life of the embryo. So we want to understand the mechanical aspects of tissue flow and this rearrangement. And the first question is, how do we begin to describe tissue mechanics? Is it liquid, right? It's flowing. Or is it, in some sense, solid? And there's every reason to expect that we'll have to go beyond textbook continuum mechanics. Because, well, I already talked about the ability of cells to divide, but there are also molecular motors that can generate internal stresses inside the cells, pull the tissue, and all of that is under exquisite control, local control, through the patterns of gene expression that I illustrated a little bit. So there is this internal information, right, this developmental program supplied by the cells. That's part of the information flow, but we also suspect that there is another information channel acting through the feedback from mechanics of the tissue onto cells themselves. All right. So let's try to think about it. Let's take a closer look at the epithelial layer, and now I'm actually showing you part of the same embryonic epithelium of Drosophila, except now you're looking not at the nuclei, but at cell and membranes, and you see this remarkably clean tiling of, so two-dimensional tiling of the surface by cells. And now let's zoom in a little. It gives you an idea of how well you can image this whole thing. But ultimately, I want to look inside the cells, and instead of showing you some super resolution microscopy pictures, I'll start drawing cartoons. So what would we find if we could look inside the cells? We would find that, well, we'd find lots of things. But let me focus specifically on mechanical elements, the cytoskeleton of the cell. It has many components, but most prominent for us is the presence of ectomysin cortex, which is made out of contractile ectomysin filaments which surround cells like little belts. And just in case, actin for us here is kind of a structural polymer, and myosin is a molecular motor. So myosins cross-link actin fibers and can walk on them, generating stress. And so that was just the cortex inside the cell. And another very important part of the story is the presence of adhesion molecules. Coherence go across membranes and form bridges between cytoskeletal bundles of the opposing cells. And so together, these things make so tissue-wide mechanical network. And right away, this mechanical network you can expect to be active. Motors play a very important role, and they can remodel this mechanical network. So, okay, so if you think that that was complicated, this was nothing like the real thing, which contains hundreds of other molecular players and complex regulatory circuits. So an immediate question is, so I, for some reason, drew that cartoon for you, and it was, you may have thought that it was already complicated, but it's nothing like the real thing, and we have to decide just how much complexity do we want and absolutely need to incorporate in the model. And of course, as physicists, we will start from the assumption that the simplest is, absolutely the simplest is absolutely the best. So in fact, already this simple model is a bit too complicated for us. So I'm immediately going to simplify it and replace this internal structure just by geometry of cells, which we can parameterize by tiling with given vertices. And we'll assume that all mechanical properties are localized here to the interfaces. That's because the cytoskeleton is localized to these interfaces. And we'll say that by virtue of the presence of these molecular motors, each interface here can generate its own private tension. And then the rest of the cell for us is going to be just the hydrostatic, in fact 2D, sort of axisymmetric pressure. Although in principle we can imagine these internal pressures varying from cell to cell. So these are, without going into any of the details of how this is generated, these are just the variables we want to introduce to describe mechanics.
Okay.
Yes?
Is this a priori justified model or a posteriori? What I mean is in physics we have separation of scales and our time scale of spatial interactions.
Well, a priori in the sense that I would argue that these are minimal local dynamical variables that you need to describe a local state. So of course you can imagine some even simpler model, just something that would govern soap bubbles. We can assume that all of these things are constant, right, and not vary from cell to cell. Right? So this is basically soap bubble mechanical equilibrium if you could go in and control the tension of each and every interface. Right? And that's where sort of biology comes in at the moment. But the rest of the biology got
do you want to assume that forces are in balance, or you want dynamics?
Thank you. I'm going there. So let's start going into a little more detail here. So I said that there are tensions here. Let's start by assuming that we can describe what's happening here by basically Hookean elasticity. So this tension is generated by a little Hookean spring, which has some internal length, internal rest length that corresponds to this mechanical filament. And we'll assume, and now the separation of timescale is coming in, we'll assume that on the fastest scale, this tissue, what it does, it comes to mechanical equilibrium. It has to balance the forces, and that's Shor's question. So let me simplify things a little further, and in fact, forget about the pressure differentials between the cells. So we only have to worry about tension, and pressure is there just to prevent the whole tissue from collapsing. So there is a tension in every edge. Mechanical equilibrium means that all these tensions have to add up to 0 at every vertex. You can do a little sweet geometric construction, rotate these tensions by 90 degrees, and then each triplet here will have to form a triangle because edges are shared between different vertices here. These triangles have to stack up, and the whole tissue then corresponds to a dual triangulation of tension space by these tension vectors.
Can I ask you a question? Is
yes.
maybe naive? This is simple in 2D.
2D.
But in 3D, that's not so obvious to do this. Why are we in 2D?
We're in 2D because it's an epithelial monolayer. And so strictly speaking, it lives on the surface of an ellipsoid. So strictly speaking, there is a little bit of puckering at each vertex, and there is a bit of a normal force, and you can imagine just getting rid of that. And so it's a reasonable approximation to the extent that the curvature is small. Okay. Where am I? Yeah. So I will, we call these things tension nets. So let's think about some properties of tension nets. You can do a little bit of geometry and convince yourself. Okay, and convince yourself. Yeah, there's an edge here missing, and I thought that this was my animation, but it's just missing. So anyway. There is an edge here, and in fact, this edge is very important, and that's why it used to be animated. In order for the triangulation, this dual triangulation, to close, there has to be one particular constraint on the angles defined by these vertices. And it looks pretty crazy. You have to look at these angles here, take ratios of sines, multiply these ratios around the plaquette, and the product is equal to 1. But it's just a consequence of the sine law. Right? You just apply sine law to all triangles. This is pure geometry, but that's actually good, because it immediately gives us something that we can test, right? The fact that there is this geometric constraint is a consequence of a local mechanical equilibrium dominated by the assumption that stresses live on the boundaries of cells, right? And it gives us something to test. And not only it gives us something to test, it gives us something to test, gives us one number to test per cell. And there are lots of cells. So we can start looking at different tissues. You see there are these tilings that we can image process, measure all the little angles here, and construct these ratios and plot the distribution of, I guess in this case, it's the distribution of the logarithm of this ratio. Right, the ratio is supposed to be 1, so then the logarithm is supposed to be 0. And what you see here is that these observed ratios indeed pile up at whatever, at 1, at 0. But then, of course, maybe they always have to pile up at 0. They do always have to pile up at 0. The statement is that the pile up is significantly larger in the observed tiling than in some suitably randomized control. So basically, if you pick angles from the same distribution but scramble them, you get a broader distribution. And since you have so many cells, this is hugely statistically significant. So there is something in that rather noisy data that suggests that there is an underlying constraint. And lest you think that this is always true, I will tell you that it is true in some tissues, but not so true in other tissues. Yes?
So what would it do if you did this analysis on foams or adhesive foams?
So we didn't do it on adhesive foams, but
In other words,
my postdoc at the time went to some restaurant, and there was kind of a stone wall there made out of cells of some marble blocks. He took a picture of that and he tested it on that. It was a much broader
no, but
distribution.
what I mean is that surface tension alone will give you a certain distribution of the vertices in a foam, which is purely based on minimizing the free energy based on deformation. Whereas if you have some adhesion, and on top of that, if you have some tension, then that should elongate certain cells, change the aspect of surface volume ratio, what have you. So I don't know how to think about how this rule changes because you have tension. Is this because of tension?
Well, it is, yes, it is because of tension and the assumption of local mechanical equilibrium. So you have to say that tension is dominated, the mechanical equilibrium is dominated by tension. So in fact, it doesn't matter that the edges are, that this tiling is a polygonal tiling. So if you take, in fact, soap bubbles, right? So then, of course, you will have circular arcs as edges. And you have to work a little harder, but there is an equivalent constraint. So, so you can describe that thing not by planar triangulation, but by triangulation which is puckered out of the plane. And that thing has its own geometric constraint.
Okay.
And yeah, you can do it there too. So, but having said that, we haven't really tested it on with soap bubbles. But there are some other tissues where you do see these sort of circular arcs, driven presumably by differential pressure. And
exactly.
That depends on pressure differences.
Precisely. Precisely.
And
So
here is your assumption that you have pressure differences?
No.
So here we're happily neglecting it. And all I'm trying to tell you is that in some other tissues, so in fact, right here, so if instead of doing this analysis, you generalize it to allow for pressure differentials, you'll get a somewhat different constraint. But it will, and that will be significantly satisfying.
On pressure differences, I think that this should be a good model. Right?
I think so.
Yeah.
That's why I asked the question.
Right.
Okay. Good. So the answer is yes. Right. So, yeah, so hopefully I just convinced you that cell array corresponds to a hidden triangulation of the corresponding tension space, right? So for a given tiling, we can construct the triangulation. And okay. But it actually turns out that going in the opposite direction, the mapping is not unique. And for a given tension triangulation, you can construct not a unique tiling, but the whole family of tilings with 1 degree of freedom corresponding to local expansion or contraction of the cell, which preserves all the angles. So this is like sort of a discrete conformal model. And so now the thought is that the space of mechanical equilibrium states, in fact, is not a unique tiling, but the whole high-dimensional manifold of tiling, which means that if you perturb the mechanical equilibrium and then you come back, you don't have to come back same place, right? So by transient perturbations of mechanical equilibrium, you can move along this manifold, right? And so we thought that perhaps biology is making use of this type of transformation. And indeed, if you look at the cells just before this gastrulation process sets into play, just before I could point, what you see, and this is running a rather short interval of time in a loop here, what you see is this deformation where you see a rather dramatic change in area of some of the cells, right? The apical area of the cells is collapsing just before this whole what's called ventral furrow is formed and internalized. So it turns out that if you look more carefully at what these cells are doing and you look at the angles, you can describe much of the variance contained in that deformation just in terms of these isogonal modes. Yes.
Is their volume changing, or are they actually shrinking, those cells? I mean, are they shrinking or are they stretching?
Let's say that the volume is approximately conserved on that time scale.
Okay.
Yeah, there is sort of 3D deformation that corresponds, and in fact, in a way, that's what's driving the change in local curvature. That so the apical area is shrinking, and so the basal area is expanding. So the cells become little cones, so they dive inside. But okay, I'm going to skip this. Okay. I'm not doing that well on time. So I just argued that this force balance constrains the shape of cells. So in fact, you can convince yourself that the geometry of cells is constrained so much that you can use the observed geometry to actually infer these internal tensions and pressures, right? So remember, we allow ourselves these variables for each edge and each pressure. If you count the number of these variables and the number of vectorial constraints at every vertex, you'll convince yourself by Euler theorem that these numbers are approximately the same. And there was a little long story about making this business of approximately the same, but doesn't matter. You can, if you forget about this for a moment, you will believe me if I tell you that there is a sort of solvable mechanical inverse problem where you look at these shapes and infer these pressures and tensions. And Madhavmani, who is a postdoc at Bell before he moved to Northwestern, did just that with some data from Tamali Kui's lab. And Tamali Kui and friends made a movie of this embryonic epithelium also showing distribution of myosin. So why is this good for us? Okay, so Manav can look at this snapshot, segment the image, and run this mechanical inverse problem, and then paint tensions on different edges. So that's the prediction. The only question is, how do we prove whether this has anything to do with anything? Because there is no easy and good way of locally measuring tensions without destroying the tissue. But in this data, we have myosin, and we're going to make the simplest assumption that local tension is just proportional to myosin. So then we can just compare predictions there to the local level of myosin, and lo and behold, the median correlation that comes out is 0.4. And it is so hugely statistically significant. So, curious situation here. You do this analysis, and you find statistically significant correlation, and now we can have a lively discussion as to whether 0.4 is a high correlation or a low correlation, and what exactly would you expect? And I would like to argue that that 0.4 actually is a rather high correlation. And in fact, it's entirely surprising that there is any such thing, because look at this. This was a movie. And I just told you that the whole thing was in mechanical equilibrium. So we made this bunch of assumptions about mechanical equilibrium. We go predict, we find correlation, but it's dynamical. So there's something for us to understand. So let's talk more about dynamics, because so far it was all static. It was very boring.
Can I ask something on the statics?
Yes.
So if you at the isostatic limit, and if you have, let's say random close packing, then you know how to go from your number of contacts or constraints to your forces from the isostatic condition, because you have the same number of equations. But in this case, if you have compressive forces and you have tension forces, is your system underdetermined? Like, is there a degeneracy in how many solutions you can find for your
No, it's
stresses?
overdetermined.
It's overdetermined?
It's overdetermined. That's the statement that, excuse me. So strictly speaking, okay, it's not overdetermined in the sense that, remember there were these isogonal modes?
Yes.
So it's determined up to those isogonal modes, right? So angles are completely determined, but there are these little breathing modes. Okay, so clearly these myosin filaments are not just hook-in springs. And one way of thinking how they're not springs is to remember that this rest length is itself a dynamical variable. And in fact, we know that if this filament is underloaded, it's going to contract. So there is reasonably well understood walking properties of this myosin. So if it doesn't carry a load, right, it will walk. There is some critical load that will stall a myosin molecule, right? That sort of corresponds to this equilibrium point here. And too much load, the whole thing will slip. And you can then convince yourself that on a time scale where these filaments start rearranging, this material can be understood effectively as a viscoelastic fluid, at least as far as large-scale response is concerned. And then the question is, Well, there are epithelial cells on my arms here, and they're not really dripping off me. Somehow we think that epithelial tissue should be able to support stress. And but in order to support stress, tensions have to triangulate. And I just told you that local tension, right, myosins on each edge have to be at stall, which is to say that in order to get tensions into equilibrium, we have to get just the right amount of myosin on each and every edge. And how do we achieve that coordination? How do myosins know what their friends are doing in the cell next door? And then you think a little bit of how you might achieve that, and you realize that there is actually a very simple way of doing it. There has to be mechanical feedback which recruits more myosin to the edge when the edge is elongating. So all that you need to do is, if the given filament is slipping, there is a signal to the cell, send more myosin. And vice versa. If the filament is contracting, that's the signal, let's get some myosin off this edge. So we call it dynamic recruitment of myosin, and it's a very simple relation between myosin and strain, except it's a very specific relation to the rate of strain, and it has some pretty interesting consequences because it ultimately says that there is no equation of state. There's no constitutive equation describing mechanics of a given given edge. It depends on history. It depends where it has been stretched or not, et cetera, et cetera. And the bottom line here is that we have 3 time scales. On the fastest time scale is elasticity, intermediate time scale, right? This internal rearrangement of internal contraction and elongation of the filament. Myosins are walking if they can. And on the slowest time scale, there is a recruitment of myosin with this dynamical feedback. And you end up with actually a mechanical system which you didn't learn about in Landau-Lifshitz, because this we certainly learned from Landau-Lifshitz, but what you have here is something that flows at relatively high frequency and becomes rigid at 0 frequency, right? So very different response. We like thinking of it as an active solid. And now the proposition is that tissue dynamics is an adiabatic rearrangement of this active solid.
Ask something. Could you go to just 2 slides back?
Okay.
No, not forward. Yeah. So you gave some plausible conveying arguments, but then you wrote a formula in the lower left,
Yeah.
which is fancy, which relates your myosin to, for some reason, logarithmic derivative
Yeah.
of your L, where from
Well,
your arguments there is
Sorry,
no.
so strictly speaking, there should be another M just to make it dimensionless, because otherwise my units are wrong. But yeah.
Okay, so this implies some strange-looking conservation law, like M plus logarithm L. Where does it come from?
I have no idea.
But why
That's a simple answer. Well,
did you not write?
well, so so this is a particular hypothesis, right? And it's a rather simple hypothesis that will guarantee convergence of this sort of thing to an equilibrium state, right? It's basically written down because maybe not on the spot, but afterwards we can go and run a Lyapunov functional for that whole thing, right? So we can convince ourselves that with this particular recruitment law, we can guarantee mechanical equilibrium being achieved at long times. But that's all that I can say in its defense.
But I guess my question is this construction unique in any sense, or you can more or less use any function more or less like this?
Well, any function more or less like this. Yeah, we can certainly quickly generalize and have a whole little class of functions that will do that. Yeah, but the non-trivial statement here is that it is proportional to the rate of strain, and that's immediately falsifiable, right? So that's what we want to demonstrate, that it actually is driven by rate of strain.
But the fact that you wrote a rate of logarithm, it's more or less arbitrary.
Yeah, just trying to
look fancy.
avoid.
Look fancy.
No, I'm just trying to avoid introducing dimensionful constants, that's all. I don't want to have to convert apples into oranges. Okay. So now the thought is, so remember I presented to you this conundrum that we were looking at cell dynamics, yet we're using mechanical equilibrium ideas and still getting somewhere. And one way of thinking about this is to think that this dynamics is happening all in mechanical equilibrium, right? So now when I drop my arm here, it's not really dropping under gravity. It's in mechanical equilibrium all the way, right? And that's sort of what I want to advocate as a view. Okay. So let's go back to the embryo. I already showed you this movie that my young colleague Sebastian Streikan made. I'm not going to play it again. Instead, I'm going to play another visualization of that same movie. So Sebastian likes unrolling the embryo in this sort of Mercator projection, where the north and so the front and the back of so the anterior and posterior poles of the embryo become grossly distorted, but we don't care. We are looking at what's happening in the middle. I'm not going to play it again, even though that should be made illegal. Instead, I'll show you the analysis of the flow that Sebastian did, just to visualize local velocity of tissue, right? So you see these little arrows, they correspond to flow velocity. If you stare at it for a moment, you recognize rather simple topology of this flow. There is, actually this is a dorsal side, and this is the ventral side, which is cut up, ends up on two edges, and the flow is converging ventrally, right? The flow is converging onto the ventral axis and is elongated along the ventral axis. And of course, if you do that, you have to have another hyperbolic fixed point somewhere else. It happens dorsally. And if you have two hyperbolic fixed points in the sphere, you also need four elliptic ones. So there are these little recirculation vortices going around. So that's the flow. Then actually, in the course of this movie, the topology of the flow also changes in some non-trivial way. So early on, there is convergence onto the dorsal side. Later on, there's convergence onto the ventral side. Anyway, so there's some non-trivial flow that we can look at. And so can we explain this flow? By taking this somewhat hand-waving analysis that I model that I presented to you, you can coarse grain it and turn it into continuum equations which relate myosin-generated stress tensor to the flow velocity, and it's basically sort of generalized Stokes flow driven by the divergence of myosin-generated stress. So a very important thing here is that myosin should be thought of as a tensor. So here is another imaging exercise. Myosin is green, so you see a lot of myosin in this embryonic epithelium. But we need to look a little bit more closely. And in this image, you can actually see that, yeah, so here you don't particularly see cells. You're just looking at myosin, but each little segment of myosin here always aligns with the cell edge, right? But then there are some edges that don't have a lot of myosin. And the bottom line here is you see that myosin locally has is characterized by presence or absence, well, intensity, abundance, and local orientation. So you describe both, and you construct this myosin tensor, and then we can talk about how this myosin is polarized, but I don't have time. So we're just going to use Sebastian's measurement of myosin tensor on the surface of the embryo and solve this little equation with two parameters. And these are going to be global parameters. We're just going to assume that there is no variation of material properties here from head to tail. And the bottom line is, using these measured myosin distributions, you generate pretty much the same flows. So here I'm just giving you a little illustration that you generate the right topology of the flow. But then, of course, topology of the flow on the sphere is constrained anyway. So that's not a very ambitious comparison. So you do a more ambitious comparison, basically comparing things locally. And the bottom line is with so 3 global parameters, you achieve of the order of 90% accuracy. And
How did you adjust the parameters?
so you just fit. But this is right. This is one parameter per image, right? You compare. Right? If you like, this effective viscosity doesn't depend on position, right? But the flow, of course, depends on position. So the only thing here that okay, there are 2 parameters. Excuse me. So
They
it's
depend
3
on
parameters. So 3 parameters because one parameter is converting apples to oranges. So you convert, right? You don't know how to convert myosin intensity into stress, right? That's one arbitrary thing, right? Which sets the overall time scale. And then another relevant parameter is the ratio of this shear viscosity and compressive viscosity that in general shows up.
But you adjust them once and for all, or you redo it, you say, per
Exactly. As a function of time, right?
But they depend on time.
They depend. Yeah. So each image, right, is right just 2 parameters. But so, in fact, there is a very there's not a lot of variation in these parameters as a function of time, but there is one very interesting aspect of that dependence. That somewhere along the way, in fact, in sort of biologically interesting moment of time, cells become almost incompressible. Right, so you effectively lose one of the terms in that equation.
Is the colored map then the error surface? Like, so the errors are larger on the ends because the theory was done on a sphere, or?
Well, the ends actually, there's just flat sheet microscopy and all the resolution. The poles goes to pot.
I understand.
Yeah. So in many ways, so the biggest errors are we are misplacing these vertices by that. But well, of course, in choosing a point to show you, I picked. So this is the error, right? I picked a good point, right? And it's not always so. So, for example, there is this 15-minute interval of time where the errors are gigantic. So, but of course, what's happening during that time is this gastrulation event. The ventral furrow is forming, and the tissue is actually going in. We never told this simulation that there was some crazy boundary condition on the ventral line, and that cells were literally getting lost and going up inside. So if you do that, by doing that, I mean if you generalize the model, of course, at the expense of more parameters, and allow cells to disappear into inside, right? So you can see here the errors piled up on the ventral surface. You can fix the problem and so recover. But anyway, I'm not particularly bragging with the accuracy of this. What I'm trying to illustrate is, in fact, once you get into the situation where a very simple model gives you a useful fit, looking at failures of the model becomes very interesting. Right? So when you see that you can't explain it in a simple way, that tells you that let's go and find something else that's happening. Okay. Okay. I'm running a bit long, but you guys were very nice and you asked questions. So, and I'm almost done. Anyway, I just want to make, I think, two little points. One is we're now in a position to go back and reconcile these static and dynamic pictures. So you realize that if you have this Myosin tensor, the equation that I just wrote down, what was driving the flow was the unbalanced part of myosin. We were driving the flow by the divergence of myosin tensor. Oops, this one. When this was constant, right? But we can add a balanced part of myosin that satisfies that, and that won't change the flow at all. But that's exactly the part which corresponds to local mechanical equilibrium. So we can go back and so subtract this from the Myosin field and redo the comparison with this geometric inference. And lo and behold, you actually find that 0.4 correlation has gone up to 0.6. So something is right, and then of course everything is still approximate. So I think that's that. And the whole story, of course, was just about, at the moment, all that we've done was related these two observables at a single time. What we would really like is to write down a dynamical equation for Myosin and be able to predict the future, not the present. And so if we could do that, right, we would be able to actually describe the flow for at least maybe 20 minutes of fly life. And then, of course, other things would happen. So the message is that these sort of simple phenological models can be quite amusing, I hope you agree with me. But what I really want to emphasize is, very often you hear about complexity, but I'm much more excited about simplicity. And I found it really surprising that we could get away with knowing so little about underlying cellular mechanics, right? Because immediately you can say, well, there is myosin, but there is also actin, and there were another 500 molecular players on that slide. So why is it that we only need to know everything about one thing and don't need to know anything about anything else? And so one thought that comes to mind is, well, it could be that there are all those other molecules, but they're just irrelevant. That they're all in excess and only some limiting step that's controlling everything. But another way of thinking about this is that, in fact, all of these things are interacting and actually very tightly correlated. So effectively, if you know one, you sort of know them all. And if that's true in this particular corner of biology, in this whatever mechanobiology of cytoskeleton, could it also be happening in other places? Anyway, we had a lot of fun with this. The experiments were largely driven by Sebastian Streikan, who was a postdoc at KITP, doing experiments at the Institute of Theoretical Physics, but now is faculty. And we had and still have a wonderful collaboration with Eric Wischhaus, who is actually a co-discoverer of this whole development, so genetic program of Drosophila development. And the theory part was done by my former student Nick Knoll and Madhav Mani and Itzi Herbskerk were postdocs at KTP, and one is now faculty at NYU, and Itzi took another postdoc, turned himself into an experimentalist at Rice, but there should be another arrow here, so now he's faculty in Michigan. And thank you very much.
Do we have any more questions?
So your last slides about dimensional reduction, can I think about it like sort of your Myosin is sort of an order parameter and everything else is slave to it?
Yeah. Well, yeah. So think of it as, so slaving is the, so I guess I said center manifold. Really, you should think of it as slow manifold. Right? And then the relevant question is, right, in physics, we're used to slow variables having to do with broken symmetries, et cetera, or conservation laws. So here, it's not obvious that it just conservation that does it. You can imagine evolution doing something like that. Right? You can actually imagine some regulatory network being driven into the state by evolution, where it allows this multidimensional but otherwise attractive manifold.
But you are saying something more, if I understood correctly. You are saying that evolution may want to bring dynamical system which a priori has all sorts of things happening on the same time scale. It wants to bring the system into a state in which one variable has much slower time scale than everybody else.
Well, no, one variable,
Well,
all
few.
the variables.
Few.
The flow in that manifold is relatively slow compared to a convergence onto it.
I don't see what you don't present evidence for that. I mean, you're just saying at the end is a few variables that control what I'm seeing here. The other one could be independent or useful for something else, and not have to be slave to these. Like if you have a slow manifold, it means that all of the other one, if I know one, I know everything else.
Yeah.
But this might not be what's happening here. Maybe there's many other variables in the system that do other things that are not related to this, right? So why does it have to be this?
I don't think it has to be.
Okay.
And if I talk any more, if I go any further on the limb, I'm going to fall off.
Any more-
It's pure wishful thinking.
Any more questions? If not, let's thank for the Thank you, everyone. Wine and cheese, 8th floor.
Yeah,
Yeah, that's what
I'm sorry, but that means logarithm
I did.
yeah.